Mr. Sarkar, you please take the presentation. Within a few minutes, we shall start our program.
রাহুল দেখা যাচ্ছে স্লাইডটা?
হ্যাঁ হ্যাঁ হ্যাঁ, দেখা যাচ্ছে দেখা যাচ্ছে।
ওকে ওকে,
আর 1 মিনিটের মধ্যে শুরু করে দেবো।
ওকে।
A very good afternoon, everybody. Today we shall start our 5th day the value-added course regarding an introduction to Ophiology, and we are here with Professor Shoikat Sarkar, Associate Professor of Zoology, Government General Degree College, Shingo, and hope you will enjoy today, which is his last class. Next 3 course, you will get 3 new resource persons, and I will also inform it in the group that in the next Saturday's class, that is 12th December, there will be a double shifting of classes, as we did not able to arrange our classes on 22nd of November. In order to compensate that, there will be an arrangement of a double class in the next 12th December. Don't worry, we shall notify it within due course of time. It's our time to have our lecture with Professor Sarkar.
Okay, thank you, Rahul. Today we'll be dealing with venom biology. Basically we'll be dealing with the infrastructure of the venom apparatus, the software and the hardware that is there in store in the snakes to synthesize this toxic potion, this deadly potion or deadly cocktail that we call venom. Now, then what is a venom? What venom is biological substance produced by organism, not sequestered from external resources. Now, external sources, say in case of certain marine nudibranchs, that means those are, nudibranchs, the zoology people will know. Nudibranchs, they sequester the nidocytes from the nidarians that they eat. Okay, and they sequester those nucleosides in the channels of their uh, GI tract, and that's how they become toxic. So that is called, this kind of being toxic is called toxic by sequestering some foreign particles borrowed from other organism. That we won't call venomous. Okay, to be venomous, you have to have your own infrastructure to produce that cocktail, and you have to have uh, a, a need to use that. That means either for predation or defense or competition, interspecific competition, whatever it is. From case to case, it will vary. Now, I mean, you have to have a program for that. Why you are using that? I mean, if you want to be venomous, it is not applicable only for snakes, for any venomous animal. Okay. Now, and the venom gland, that is the factory that will produce the cocktail for you, and the delivery system. It can be injection, it can be a spark, whatever it is. The delivery system must be there. I mean, injection means not the kind of injection that we talk about. You have to have a structure that will directly inject the venom or introduce the venom in the targeted victim. Now, what are the characteristics of animal venom proteins or animal venoms, you may say? I mean, it is not 100% protein, but principally it is proteinaceous. All the venoms are principally proteinaceous. Okay, say 70 to 90% you can find protein there in many venoms. Now, at least in snake venoms, you can find 70 to 90% protein. Okay, now co-opted or recruited from physiological body protein encoding genes. Now, these venom candidates, or the toxin candidates that are components of the of any venom cocktail. These are not foreign genes. These are our own genes. These are their own genes. Now, these genes are co-opted. There are mechanisms. There are 4 or 5 mechanisms by which actually these venomous animals co-opt the genes. They can duplicate and they use it. I mean, they express those genes in the venom gland, and we get a cocktail. Okay, so that is called co-option and recruitment. The genes are there in every cell, but they are co-opted to get produced or get synthesized and secreted. Not as we know, we are genomically equivalent. The eukaryotic organisms are genomically equivalent. Means all the genes or all the cells of our body have got the same contingent of genes, but not all the genes are getting expressed in all the cell lines or all the tissue systems that we have. Like we have melanin in our hair, but it is not getting expressed. I mean, the melanin gene is there in your hair follicles, in your heart, in your heart cells, in your lungs, whatever tissues that you have, everywhere in all the genes that in all the cells that we have the same gene. Okay, but this melanin gene is not getting expressed everywhere. So genomically, we may be equivalent. All the cells are having the same contingent of genes. But proteomically speaking, all the tissue of our body, they are not equivalent. Okay. Different patches of genes are getting expressed, getting selected to get expressed in different tissue systems. So that kind of co-option, gene co-option, is happening in case of the venom gland. Some of the genes, normal genes of their body, is getting selected to get expressed in the venom apparatus or in the venom gland, and this added secretion that say C-type lectins or any complement protein. The complement protein, it is our physiological protein. Now, you do not know if you start excreting this or secreting this particular protein from your salivary gland, then you may become venomous. So that is called co-option, gene co-option. It is not a very normal site to get expressed, but this kind of abnormal co-option or unnatural co-option sometimes can turn a normal animal into a venomous animal or a toxic animal. Okay. Now, most toxins are secretory proteins, as I just said. Uh, the majority of toxins are cysteine rich. These are the trends. These are the general rules. There are variations, but these are general rules. Many toxins undergo post-translational modifications. We know about there are different kinds of post-translational modifications. There is sulfation, there is what we call glycosylation, as you know. Uh, uh, Different kinds of post-translational modifications very often happen with these toxin candidates, and these modifications actually we call it tinkering with the basic infrastructure or basic scaffold. The protein is the basic scaffold, and these modifications in many times in many cases, these post-translational modifications actually turn a simple protein into a very toxic component. Okay. Now, variability in the evolutionary rates of toxins. This is very common. Uh, you have got 10 toxins in a venom, maybe. Now, out of those 10 toxins, maybe any three are changing very fast, but it is not applicable for the rest seven. Okay, so if you have got 10 genes, 10 toxin secreting genes, I mean the rate of change or rate of that we call evolutionary rates in variability, that may differ from one toxin to other. Now, differential evolution of domains even. Say you have if you talk about three finger toxin. Now, three finger toxin in one snake, maybe one. We have got three domains. Three finger means three finger domains. Okay. Now these out of these one, two, three fingers, maybe finger two is changing faster than the other two. Okay. So there is a difference in the rate of change of domains in a single protein also. Okay. So these are rules. And of course, venom evolution is largely driven by species ecology. That is a very simple rule. It has to be connected with some kind of advantage, ecological advantage, otherwise it won't be selected by natural selection. Now, talking about the composition, as I just said, that most of the venoms are actually proteinaceous, protein rich. These are protein rich potions or protein rich cocktails. Up to 70 to 90%, it is proteinaceous, and it is a cocktail of enzymes, non-enzymatic components, metals, amino acids, even nucleotides are there, carbohydrates, lipids, biogenic amines, you name them. Just, I mean, this is a system to dislodge your opponent. So all kinds of make a very efficient, make your aim should be to make a very efficient cocktail that will in one way or other pull down your competitor, whether it is a predator or a prey. Now, the biological composition of venom toxins among snake species can vary greatly. It is a very obvious thing. The kind of venom toxin that we find in case of vipers, obviously they will not be there in the same amount or with same efficacy or seen in the same nature in elapids. Okay, so from snake species to species, it will vary. This is very normal. Although in general, the dominance of major protein families in venoms often follows a broad taxonomic trend. That is also true. That means, say, vipers. Vipers are a group of snake. Elapids are a group of snake. So is there any trend in the composition? Yes, there is trend. In elapids, if you put all the elapid venoms under analysis, and if you analyze all the viper venoms, all the crate venoms, you will find a trend in the group. So there are certain common trains in the crates. There are certain common trains in cobras. There are certain common biochemical composition trains in elapids as a whole group or viperids as a whole group. We will discuss. Okay. Now this is the broad taxonomic trains in venom composition. That, I mean, the picture that you are looking at. It is now this is a study done by these two fellows. Now elapids, 42 species, with 42 species of elapids, they have done this study. But this is largely, I have tested it, I have checked it. I mean, the species that were not included in this particular study, I've gone through their venom profile whenever it was available to me. But the almost the same trend is followed in those species also that were not treated for this particular experiment. This is a fantastic study. Okay, now they have done this experiment with 42 species. They have done this analysis with 42 species. That means the venom analysis. This is the pie. This is the pie of venom pie chart of venom analysis. Now, as you can see, here you are. I mean, the principal three components are phospholipase A2, SVSP. That means snake venom serum protease. Yes, serine protease and snake venom metalloproteinase. Phospholipase A2, that means the first three components. And there is three-finger toxin. Three-finger toxin you won't find in vipers, but these three you will find in elapids as well as vipers. Now, this is the general elapid trend. This is the general viperine trend. That means Viperini subfamily. There are many viperine species, but they have taken only 20 out of them. Now, here you can see it is laden with SVMP, metalloproteinase. Okay. Now, in case of elapids, you can see it is laden with three-finger toxin. I mean, the principal share of venom pie is three-finger toxin. Now, if you consider crotallines, you can find there is good amount of both SVMP as well as SVSP. So these are some common traits. There are deviations, there are exceptions, and there are some unique venoms also which do not follow any of these rules. We will discuss. Now, talking about the general trends. These are the general trends. Okay. Now, in writing, I have compiled that. Now, FFCs, that means prawn fang xenophidians, contain approximately 59 protein families. More than 90% of elapid and viper venom composed of just 10 protein families. Now, of these 59 protein families, most of the viperids and elapids have a lion's share of their venom composed of toxins from 10 protein families mainly, and other 49 families are not the dominant ones. Okay, they are the minor ones. Now, these 59 protein families are classified into 5 groups. Group number 1 is dominant protein families. That is, there are 4 families of proteins there. Phospholipase A2, SVMP, SVSP, three-finger toxin. Yeah, I have shown you just now. Phospholipase A2, SVSP, SVMP, and three-finger toxin. These are the dominant ones. You can find it everywhere. Now, secondary protein families are Kunis, Krebs, Lau, CTLDs, and NP. NP means natriuretic peptide, disintegrine, C-type lectin, L-amino acid oxidase, Krebs, Kun means Kunis. Now, these are the secondary protein families. That means they are not the principal ones, but I mean, there are certain species or there are certain groups of snakes where you can find some of these, some of the representatives from these families are there in good amount. Okay. Now, this is there is again minor protein families. These are the representatives of the minor protein families. There are rare protein families. There is 36 in that group, and unique protein families. defensins, viglarine, matigotoxin, cystatins. These are examples of that fifth group. Now, these families make up 38% of the whole venom of single species. I mean, the species that is having any of these things, I mean, maybe you can find one species, say, talking about Waglerine, there is Tropidolemus species where you can, I mean, Tropidolemus wagleri, temple pit viper, where you can find these Waglerine, I mean, it is having the principal chunk of the pie, of the venom pie. Okay, so, but it is not having defensive, it is not having metigotoxin. Okay, in some other species, in a coral snake, in particular type of coral snake, you get this particular component, that is metigotoxin, I mean, getting the large chunk of the pie, venom pie. Okay, so this kind of, I mean, these are called unique protein families. That means these are not present in most of the snake species, but wherever they are present in good amount. Same thing, defensins in Crotalus, Waglerine in Tropidolemus, metigotoxin in Calliophis, cystatins in Bitis. These are those rare families. These are representatives of those fifth group of toxins. Now, talking about cystatins are present in Bitis, in most of Bitis, not all of them. In most of Bitis, you can find cystatins. So this is unique. And there is also one group from coral snake that is having cystatins. I mean, these are poles apart. These are some of those freaking things that you can't explain all the time. Okay, so the cystatin is present in Bitis. It is a viper, and it is also present in Calliophis. It is a coral snake. That means elapid. Okay, so these two snakes are poles apart, but still you are getting the same component. That means separate recruitment events of some physiological gene has taken place separately in Bitis as well as in Calliophis. Now, let us concentrate on elapid venom profiles. Now, this is also coral snake, same genus Micrurus. This is also coral snake, same genus Micrurus. But you are looking at a group of Micrurus species which are having three-finger toxin as their dominant component. This yellow share is the three-finger toxin. And you can find some other species under the same umbrella called Micrurus, where you will find phospholipase A2. Phospholipase A2 as the major component. Okay, so in the same genus, you can have species which are having absolutely different protein profiles or absolutely different toxin profiles in their venom. Now, coming to the Australian elapids, we have trains. Trains like, there are mildly venomous ones. I mean, most of the Australian elapids, at least terrestrial elapids of Australia and Papua New Guinea, most of them are medically important. There are also some which are not that much medically important, but still they are elapids and they are venomous and can kill you sometimes. Okay, not always, maybe. Now, in those mildly venomous Australian elapids, you can see the large chunk is taken by three-finger toxin, whereas in case of medically significant Australian elapid, these are the trains that I'm talking about. In case of medically significant Australian elapids, you can see the phospholipase A2 is getting the lion's share in the venom pie. Now, coming to the cobras, as you already know, there are 33 species of cobras, but here in this study, they have done this study with 11 species only. And you can see here, three-finger toxin is the main component. You are having quite a and some amount of phospholipase A2 also. And in crates, you're having phospholipase A2. Now, talking about this phospholipase A2, this is phospholipase A2 in vipers. You can have vipers with the lion's share in their venom pie with phospholipase A2. Now, that phospholipase A2 is different from this phospholipase A2. Same protein uh, scaffold, but performing absolutely different function. The mechanism of action is can be absolutely different. Okay, I mean those tinkering events. That means those post-translational modifications, those tinkering with the basic uh, scaffold of the protein or basic scaffold of the molecule can immediately introduce some kind of new functionalization, new functional attributes to the molecule. Okay. Now this kind of say this is a phospholipase. I mean, in case of cobras, you can get phospholipase A2. You can in case of crates, you can have phospholipase A2, and in case of vipers also you can have phospholipase A2. But there are cobras and there are vipers where you can have these phospholipase A2 as highly cytotoxic components, and in case of crates, the same phospholipase A2 is highly neurotoxic. That means the mechanism of action is absolutely different from some of the NASA and some and all almost I mean majority of vipers. Okay. Now coming to black mamba, this is a unique species because here the principal component is cunids. Cunids is present in crates also, and other elapids you can find cunids, and in some vipers you can also find cunids. But cunids is the main component. It is only applicable for black mamba. In no other snake species you can find this kind of pie. So this is a unique scenario. The same thing that I discussed just now. Those are written in words only. Okay, you can go through these things. Say in about 90% of elapids, greater than 75% of the total venom composition is made up of just two protein families, phospholipase A2 and three-finger toxins. True. I discussed, I have shown it just now. The venoms of medically important Australopapuan elapids are having phospholipase A2s, and the less important ones are having three-finger toxins. Okay, I mean three-finger toxins as their principal component in the venom. Now, all the most basal species in every clades. Now, coming to the evolutionary aspect of snake lineages. Now, if you consider the evolutionary aspect of the Australopapuan lineages of elapids, you can find the most basal species are having predominantly three-finger toxin in their venom. Whereas the most, I mean, the derived ones, that means the later ones, most advanced ones, are having phospholipase A2s as the principal component. Again, a very important trend. Okay, now crates, definitely phospholipase A2 is the principal component, or it is having a lion's share in the pie, uh, uh, making up almost half the venoms with less three-finger toxin, similar to the medically important Australian elapids. But there is a difference. This having high amount of phospholipase A2 in the Australopapuan elapids, terrestrial elapids, and in crates, the mechanism of action is different. This is highly neurotoxic, and many of these, I mean, these species, which are they are neurotoxic also, but equally they are equally some of them are actually highly cytotoxic also. Okay, but this phospholipase A2 is almost 100% it is neurotoxic. I mean the crate ones. Now crates contain larger amounts of secondary protein families. Just I discussed cunids, laus, crisps. So crates are different. This is a different lineage among elapids. All the elapids are having some kind of common trains, but the crates defy those trends. So these not very principal components are in much more amount in crates than in other elapids. Now talking about the elapids with unique venoms. Let us talk about some elapids with unique venoms. Calliophis, Dendroaspis, uh, mm, I mean, apart from Calliophis we already know that they are having those marigotoxin in their venom. But Dendroaspis, we just discussed, it is also having a very toxic venom. I mean, a very unusual venom. I mean, it is having huge amount of conids in it. There in Dendroaspis, there is absolutely zero phospholipase A2, but huge amount of conids. Three-finger toxin modified into both acetylcholinesterase inhibitors. I have got a picture for that. Now, three-finger toxin modified into both acetylcholinesterase inhibitor as well as L-type calcium channel blockers. Same toxin family, three-finger toxin, but having two different functions. Absolutely two different functions. Now, additionally, the conids are present in Dendroaspis in far greater amounts than in any other elapid. Other elapid species are also, and a few vipers also have conids, but other elapids that are having conids, they are having it in very less amount, unlike Dendroaspis. Okay. The most apparent venom displayed by any elapid species is, I have written it in bold, is Malayan blue coral snake, that is Calliophis barbagata flaviceps, which possesses the highest amounts of aspirin guanin of any elapid species. So this is very unique scenario. These are those minor families of proteins, or you may say the unique families of proteins. Okay. Now, unusually high amounts of HBMPs in Calliophis. Generally, the vipers are having huge amount of HBMPs, not the elapids. But as an elapid, they are having very huge amount of HBMPs. And the principal component of their venom is maticotoxin A. It is a typical cytotoxic thing. And the Calliophis bivagata is a snake-eating snake, that is Ophiophagus snake. And they specialize in predating on the most toxic species, sometimes baby king cobras also. Now, the highly aberrant venoms of these genera, that is Calliophis and Dendroaspis, could indicate an ancient phylogenetic divergence. This is also very natural because their venoms are so very unique. I mean, there are so many unique features in their venom. It seems that they have diverged from the rest of the elapids quite early. Now, there is pronounced geographical differences in the trains also. We have discussed, we know something about, we know the term that is geographical difference in venom composition. It is common, but there is also a trend in that. Now look at these microdus species. There are two color codes. Okay. Now these microdus species, as you move from this Mesoamerican region, from this Mesoamerican domain to the North America, you can see there is more of phospholipase. I mean, these venoms of these microdus species are having more of phospholipase A2 than the venom of the microdus species that are found in South America. So there is a clear trend. The South American microdus species are having more of three-finger toxin in their venom pipe, and the North American, not very north, but close to Mesoamerica. Now these North American species of microdus, they are having more of phospholipase A2 in the venom pipe than three-finger toxin. So this is a very clear trend. And if you investigate the same train, if you try to find whether the same train operates in our case also, in India also, I'm sure you'll find it. It should be a global phenomenon. Now let us concentrate on viperine venom profiles. Don't get nervous. These are, I mean, there are trains. I'll talk about only trains here. Okay. Now, as you can see, in Daboya Raseli, that is our Chandrabora. In Bengali, we call it Chandrabora, or Russell's Viper. You have got unusual share of purids. Unlike other, other viperines can also have this Kunitz peptide, but not this amount. This is unusually high as a viper, even much more than many elapids. Okay, so this is a very unique scenario. And you can find huge, these phospholipase A2, SVSP, SVMP, they are taking as a group, they're occupying the lion's share almost everywhere. So these are the trends. In H's, you have got SVMP as the principal component, and SVSP is very less as compared to other vipers. In H's, that is soft scale viper, you are getting a very huge amount of SVMP share in the venom pipe than any other vipers. Same thing. If you go through the slides later on, you can find similarities, okay, with these slides and the picture that I have shown. And yes, in case of viperate venoms, pharmacologically speaking, the viperate venoms are largely, not all of them, but largely they are hemotoxic or histolytic in nature, as opposed to the elapid venoms, which are principally neurotoxic. But as I said already earlier, especially the elapids, the cobras, African cobras, which are mainly venom squatting ones, that is spitting cobras, they are having principally histolytic nature of venom, unlike other cobras, which are principally neurotoxic, having neurotoxic venom. Okay, but the viperids, as a rule, they are having histolytic venom, that is myotoxic, hemarigins, hemotoxins, cytotoxins. These are the principal components of the venoms. Now, lachesis, it is an American species, Lachesis muta, has significantly high amount of natriuretic peptide in its venom. Mesoamerican species, these are some examples of the Mesoamerican species, all possess noticeably large amount of these components, unlike other Mesoamerican species, or unlike elapids. Unlike other viperids of that region. Now, the most aberrant venom of any viper is Tropidolemus, that is the temple pit viper that I was talking about. In addition to having 38% of the venom being represented by a unique family of Waglerin, three dominant protein families in other viperid venoms, that is phospholipase A2, SVMPs, and SVSPs, made up only 15% of its venom proteome. Proteome, you understand? Proteome means the total number of genes that are getting expressed in a particular tissue, maybe. Or if the background leaf, or if the scale is the organism, then we call proteome of that organism. If the scale is a tissue, then we call a proteome of the tissue, like that. Okay. Now, 15% in this particular Tropidolemus, the major players, and these three are the major players in other venoms. Now, these three major players are actually the minor-most players in this particular venom, where Waglerin is the principal player. He is the captain. Now, this is about origin and diversification of toxin genes. What are the mechanisms by which the genes, the physiologically important genes, are getting co-opted, and after certain changes, they are acting as toxins? What are the mechanisms by which we transform a docile physiological gene into a very hostile toxic gene, okay, or toxic component? Now, this is, as I say, this is a physiological gene. There are events, duplication events. I mean, gene duplication. You know, all of you. Now, these gene duplication events in, I mean, before new functionalization, we call it new functionalization. Before gene duplication, I mean, before putting it under the natural selection, there are events of gene duplication. That means the physiological gene is kept intact, but we are just copying the physiological gene, and all those copies are put under selection pressure in the venom gland. Okay, now there is rapid evolution under positive selection. If they at all work, fine, then they are positively selected to act as toxic components of the venom, or they may turn into, I mean, because of those mutations, it may turn into pseudogene also. That means non-functional gene also. That means this gene has failed to act as a toxin component. Now, we know about transplicing, we know about alternative splicing. Means our gene, those who are not from biological background, the genes that we have, that is getting expressed in as heterogeneous nuclear RNA. This heterogeneous nuclear RNA is having two components. One is called exon, another is called intron. These intron parts are spliced out of the final product, and what we get after the splicing event is just compilation of the nearby exons. So that we call splicing. Now, in alternative splicing, some of the exons are also getting kicked out of the final product. That is called alternative splicing. Say we have got 5 cassettes of exons in a particular gene stage. Now, in one product, we are having, I mean, as final product, we are having 1, 2, 3, and 5 exon. I mean, the first, second, third, and fifth exon in the final product. In another, I mean, after another splicing event in some other tissue, maybe we are having 1, 2, 3, and 4, not the 5 in the final product. So that we call alternative splicing. There is transsplicing. That means one gene segment is attached to another gene segment. That is called transsplicing, and we are getting the final product after that. Now, there is new functionalization. That means after tinkering with some domains of a particular scaffold of a structure, then we can find an absolutely new function. And because of this new function or new attribute to the functional attribute to the scaffold, one simple gene product, one non-toxic gene product can certainly become highly toxic. Okay, and if it is selected, if this new functionalization is selected, there is repeated duplications, and due to this repeated duplication, a gene, a very normal gene product can become toxic. I mean, this is the seventh mechanism by which a simple gene product can also become toxic. আমরা যদি কাশির ওষুধ বেশি করে খেয়ে নিই, then we will fall ill. Okay, the cough syrup that is there in the market, if you take 2 vials of those cough syrup, you will die. Okay, so simple products can turn into toxic products if you take too much amount. So these are all those 7 steps, 7 mechanisms, 7 ways by which you can turn one simple product into a very toxic one. Looks can be deceiving. This particular proverb you have seen earlier in case of snakes. Now it is applicable in case of molecules. These are the two sides of the same protein. Okay, 180-degree different. But look at the next pic. All of these are three-finger toxins. These are only 180-degree different. That means these two are same molecules. Now, but these are different three-finger toxin families. All of them are under toxin families, but these are different subfamilies of three-finger toxins. Now, in this family, it is acting as sodium channel activator. Family 2, three-finger toxin, is acting as calcium channel blocker. Family 3 is acting as anti-acetylcholinesterase. They are acting as acetylcholinesterases. Family 4 is acting as muscarinic acetylcholine receptor antagonists. That means they block that receptor. This is nicotinic acetylcholine receptor antagonist. That means they block nicotinic receptors. All of them are three-finger toxins, but they are performing absolutely different functions. So, tinkering with the basic scaffold of a particular protein family, you can have very different functions. Okay, and these are the office sites. These are the office sites of these proteins or of these toxins. This is the office site of sodium channel activators. This is the office site of calcium channel blockers. So the office site means these are the regions where they act. Okay, and by acting like that, they are actually interfering with the functioning of the neurons. Three-finger toxins, they principally inhibit or make our nervous system dysfunctional. Okay, in many ways. So these are those many ways. Three-finger toxins, these are those 5 ways by which the different groups of three-finger toxins can actually knock down our nervous system. Same thing in words. Now, the infrastructure. Till now, I was discussing about the software part of the infrastructure. Now, let us talk about the hardware part. Now, this venom apparatus, as you know, in the venom apparatus, you will be having a gland, you will be having a venom delivery system, and of course, the potion. And the potion, and for synthesizing the potion, you have the software, that is the genes. Now, the genes, they are giving you the product, that is the toxins, and all the toxins come out of their synthesis sites and make a cocktail which we call venom. Now, this is a venom gland. As you can see, this is maxilla and the fangs. These are the fangs, and the fangs are at the rear end of the maxilla. That means this particular snake is an opistoglyphus snake. Here you can find the maxilla, but it is a fixed one, and you are having a fang at the front end of the maxilla. So these are proteroglyphus snakes, but these are erect fangs as opposed to these ones. These are foldable fangs, not foldable fangs. Their maxillae is actually very highly mobile and they can fold. It looks like the fang is fold, but actually the maxilla is fold here in horizontal position. These are the variations in the structure of the gland. You can have a very fuzzy structure in case of the Duvernois gland of the opistoglyphus snakes, as you can see here. This is the venom gland. Now we call it venom gland, but earlier we used to call it Duvernois gland. All the opistoglyphus snakes, we used to call them having Duvernois gland. Okay. Now they have got a very short common duct, and this is the parenchyma. That means the main parenchyma of the gland. That means these are the structures. These are the areas where the toxins of the venom are getting synthesized and secreted through the duct. Now, look at this venom gland structure. It is different from this structure, and you are having a synthetic area here, and you are having a transport area here. The duct is at the anterior end. It is not coming from the rear end of the entire gland. The duct has originated from the mid portion of the gland. So this is the synthetic part. These things, I mean the products, all the toxins in the venom cocktail will just flow through this duct and will enter the anteriorly placed fang. Now talking about the venom gland structure, clearly different. It is a very big venom gland with respect to its head. The venom gland of the viper is quite big, and these are the cords, these are the tissue cords. That means these are the factories where the cocktails are getting generated, or the different toxins are getting synthesized and secreted by different groups of cells in these cords, and together, they come up as a cocktail and flow through this duct. We are having an auxiliary gland here in case of vipers. Now, in case of attractospids, we are attractospidids. We are having this kind of a gland. The gland duct is going till the rear end of the gland. So these are the variations. These are some of the variations in the venom apparatus or the infrastructure for venom delivery in different groups of snakes. Now, we use this term spitting and non-spitting snakes, specifically spitting and non-spitting cobra. Now, what is the difference? If you look at the anterior end of the, or if you look at the tip of the fang of cobras, now in case of spitting cobras, you will get a very small orifice through which the venom is coming out. But in case of non-spitting ones, you'll be having an elongated appearance of the venom aperture. The venom aperture is quite elongated, 2-3 times more elongated than the venom aperture that you find in case of spitting cobras. Okay, I mean this small size of the orifice actually helps the snake to create the jet of venom that is thrown at you. Okay, so that is the difference. The ones with an elongated orifice are non-spitting, and the fangs that are having a very small orifice at the tip are likely to be spitting. This is the direction of venom flow through the fang. As you can see, it is a non-spitting cobra. It is going straight. In case of Naja, a slight tilt you can find till they are not, still they are not spitting. And in case of Naja nigricolis, they are clearly spitting cobra. In case of Himachaetas, they are not cobra, but they are spitting. They can squirt their venom. These are called wrinkles. They are good spitters. Okay, so and this is the, why this snake cannot spit. This is the interior of this is the interior base of the fang, the cross section of the fang. The venom is coming through this canal, and there is no deviation here. So the venom will go straight like that. So they cannot squirt the venom. But in this case, just at the base of the orifice, you can find a deviation. Okay, now the venom is coming very fast through this canal, and through this orifice, it is going straight in 90 degrees. So it is a spitting cobra. The fang of a spitting cobra. Poisonous snake. Yes, I'm right. I'm not wrong. There are venomous snakes as there are non-venomous snakes. Plus, Plus, there are poisonous snake also. Being poisonous and being venomous, these two are not the same thing. Venomous means you have to have, you already know what is a venomous snake. You must have a dedicated infrastructure to introduce, to synthesize and introduce the venom into the target organism. You are not allowed to sequester some other toxin, some other animals toxin and use that for then you won't be called venomous. Okay. Now poisonous. What is a poisonous animal? This is basically talked about in terms of trophic way of getting the taste of the portion. Okay. Now if some predator eats this snake, it is likely to die, as because it is poisonous. The snake has not bitten it, but the snake has something in it which is toxic. If you eat it, you will die. Okay, there is no need for the snake to bite you to kill you. Then we call that particular snake poisonous. Now it is this structure, we call this structure nuclear gland. This is the battery of those sequestered toxins. I would call sequestered toxin because trophic means they gather these toxins from their prey items, the particular type of bufonids that they eat. From there, they actually sequester this toxin, and some unknown means they are able to actually sequester those toxins into these batteries of, into this battery of glands, you may say, or storage spaces. Okay. Now, whenever any predator approaches this particular species, this is called Rhabdophis tigrinus or tiger snake, not tiger snake, tiger rat snake, maybe. Rhabdophis, the scientific name is Rhabdophis tigrinus, and it is having this kind of a structure, and it is full of toxin. Now, the moment a predator approaches it, it just sways its anterior part of the head towards the predator, so that the predator bites this area and dies, or at least stop biting, because this is a very toxic component. Now, what is that component? These are bufodienolides. These are cardiac glycosides, and this is the portion. This is the toxin. This is a toxic cocktail. Okay, it is coming out of the gland by bursting those glands, and again the structures will form. Okay, now these are nothing but cardiac glycosides, very toxic components, and these are the effects of coming across this particular toxic component. This is atrioventricular block, bradycardia, ventricular tachycardia, lethal cardiac arrest. These are the effects of this, different grades of effects of this toxin. And as I say, this is the nuclear gland system. And what are these nuclear glands? The organs which superficially resemble secretory structure. They do not actually secrete. They do not have a canal or channel to secrete these things. Okay, so these are artificial. They're not. They look like glands, but they are not actually. Excretory. Secreting anything. Okay, embedded in the dermal layer of the dorsal skin of the neck. The nuclear glands or Rhabdophis tigrinus contain cardiac. Whatever I said just now. The glands of some other species also. These are not the only species that are having nuclear glands. There are a few more species of snakes that are having this kind of structure, and they are also able to sequester bufodienolides. Now, ontogenetically, the nuclear glands are mesodermal in origin. This is very important for your exam also. These, unlike other, skin glands, these are mesodermal in origin, not ectodermal in origin. The glands lack secretory epithelium. They do not secrete. They do not synthesize or secrete anything. Though we call them glands, they do not have any secretory epithelium, as because these are not their products. These are the products, or these are the components that they have sequestered from other organisms by eating other organisms. Okay, so these are the glands that actually do not have any secretory epithelium. No central lumen. There is no central lumen as other glands have. There is no duct. Glands simply rupture. These we should not then call them glands. These are those structures that get ruptured when you manhandle them, and the toxin comes out, and you die, or the predator that tries to eat it, it dies. So I think that will be all for today. These are some of the references, not all of them. These are some of the references that I have considered to make this presentation. Uh, Thank you.
Thank you, Shoykot.
Hi.
Now the audience are requested to put their questions either in the chat box in both YouTube as well as this Gmeet, or they may directly ask the questions to Professor Sarkar.
আমার মনে হয় language is a barrier here. Anyway, let's see how they perform in the exam.
That should not be, because the people, all of them are well understood in English, and if any problem is there, they are free to ask the questions.
Yeah, in any language, if you have any
language বলা আছে,
একদম
না বোঝার তো কারণ নেই, এত সুন্দরভাবে স্লাইডে বলা আছে, লিখে দেওয়া হচ্ছে। যাই হোক, আমি চ্যাট বক্সটা ফলো করছি। আমি ইউটিউবেও কোনো প্রশ্ন দেখতে পেলাম না। I have not find any questions in YouTube also. আমি আরেকবার দেখছি মেসেজে। Yes. Some questions are there. What is the most venomous snake and what is the most poisonous snake in the world? One question.
The most poisonous snake is this one. The poisonous snake is this one. The snake that you have just seen. This is the most poisonous snake. The most poisonous snake. The most venomous snake will be fear snake that is found in Australia. Fear snake or inland taipan.
The next question. The previous question was asked by Shagota Koel. Moinak Mondol is asking, all the splitting snakes must have solonoglyphal fangs. আচ্ছা, প্রত্যেক সবকটা স্প্লিটিং
No. Spitting. I mean, spitting. Till now, we have only one viper species that is believed to have been seen to squirt some amount of venom when manhandled. That is Mansion spit viper, but that can be a myth also. Okay. I have not seen it. I've seen so many YouTube videos of Mansion but never I've seen it spitting. But it is said that they can spit sometimes. Okay. But spitting is unique in some cobras and in wrinkles. Wrinkles can spit. They are actually better spitter than many of the bona fide spitting cobras. So some cobras can spit, wrinkles can spit, and of all the vipers, these Mansion spit viper or Mansion viper, they are believed to be spitters.
আচ্ছা, সোমশুভ্র দত্ত is asking that is the geographical variation in snake venom depend on their prey species? If certain prey species are somewhat resistant to venoms, okay. Is the geographical variation in snake venom depend on their prey species? if certain prey species are
Yeah,
somehow
yeah,
resistant to venom.
yeah, 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 yeah. Of course, because this is the same thing. I mean, this is a treadmill. The prey predator relation in terms of ecology, they are always running on a treadmill side by side. Now, to keep on the same place, you have to run all the time. Alice in Wonderland, if you remember, to keep in the same place, you have to run all the time. That means if you do not change, at least your prey as a predator, if you do not change, then you will fall back. Your prey will be advanced, and you will die. You will become an extinct species. So, as your prey item is trying to make shields against the bullets that you produce, you have to create advanced bullets that those shields will fail to deter. That means your prey item is changing their molecule. Your prey items, if you are a snake, your prey items are always changing. They are also trying to live. So they are changing their protein profiles. Now, if you do not change your armory, your molecules, then you will be extinct. So as the prey items are also changing, so you have to keep in the same place at least. You have to change all the time. So yes, with the species, with the prey items, as they are changing, the predators also change structurally as well as in molecular terms.
Well, Minak Mondal has given the thanks for the answer, but no question, neither chat box or
ইউটিউবে কোনো কোয়েশ্চেন আছে?
ইউটিউবে তো বসে আছি, শুধু 4টে লিঙ্ক দিয়ে বসে আছি। ইউটিউবে তো শুধু 4টে লিঙ্ক, আর কিছু দেখতে পাচ্ছি না।
ঠিক আছে, there is no question, that's okay, then call it a day.
Okay, we shall close our session today. Any question, please? Is there any question? কোনো প্রশ্ন আছে? So I think Shoikat, it is time to close the session.
Okay.
We shall
Thank you.
the next Saturday. Please do remember that in the Saturday, the time will start from 5 p.m. as usual, but it will continue for 2 hours because that will consist of 2 lectures. এই সামনের শনিবার যে ক্লাসটা হবে সেটা দুটো ক্লাস হবে, 2 ঘন্টার দুটো সেশন. 2 resource person will talk in 2 different topics, and in the next Sunday you will have your exam because you have to pass in the exam to get the certificate from this course. Okay, so I think this is the end of session.
And for the exam, I'm sorry to interrupt. And for the exam, the slides, you have to go through these slides very carefully. The questions will come from these slides, and the Word document that I have shared with you in the group, also go through that. Okay, the questions will come from these slides as well as that doc. Thank you.
Okay, so let's close the session. We shall meet in the next Saturday. Thank you, sir.
Thank you, Rahul. Thank you.
Hello.